So, and for those who have not had me, and some of you have, and some of you haven't, to be fair, uh, I will. I've gotten into the habit with COVID of recording all of these things and distributing them. So I already send. I convert them to YouTubes, and I've been sending them now rather than putting them on the site. I'm just making them as emails, and so you listen again, and frankly, I think listening more because you can always listen again and do notes. I'll try to point out things that are important, that kind of stuff. So I do that routinely, and that's why when I sent you the invitation, I mean, it's up, look, if you don't want to accept the invitation, that's fine. The only thing that is if you do have to be remote, then I have to ding you in if you're not already on the list. But that's okay, you know, depending on how things go. So you'll see it here, and that's this thing. So you're E, and even though we'll cover the same material, this class is a little more crowded. We have the better part of 30. They have about 22. So it's slightly different. At least it's in the same classroom, which makes it easy for me with all the materials already loaded that are on there. So first part. Again, I don't want to spend a lot of time. Syllabus micro for if you're not in micro party, you're in the wrong room. But by this time, I suspect everybody knows where they're supposed to be, and that's me. And so the usual, I'm only on campus for the most part between on Monday, Wednesdays, and Fridays. So I generally just hang out at my office over that John Jay, the main building, which all of you will get to know because you have lab over there. Okay, your micro lab is over there. Except rather than going to the micro lab, making a left and then a right and going straight back to the lab, if you go straight back, we call it the atrium because you enter there. This level is the second level, the gym level of John Jay, and all the science offices and some of the engineering offices are there. The atrium is an open area, and my office is sort of toward the right corner. And so typically I'll be, hanging around there, or somebody will know where I am. And other than that, it's easy enough to construct virtual meetings if you would prefer that. We respond to emails very quickly, as many of you already know, and that's that part. Lectures, because some of you have me for lab, some have me for lecture and for lab, depending on the courses that we became acquainted with, and those haven't had me before. I'm very much exam oriented. Okay, there's no projects, there's no homework. Like that. Take exams. This is the way we've constructed. Now, other instructors may do it differently. Other folks who have micro who are teaching it, and that's their privilege to do that because I have two, I guess, of the three sections. You tailor the course. We have a course called 2390 or General Microbiology, which is for the majors, for the bio majors and pre-meds. They get nothing clinical. They learn very little, if anything, about diseases or anything like that. This course was constructed originally, and the people who took it the most by far and away were nursing and health science related students. That's the part I like to teach because my background is clinical. Okay. I took care of people for 25 years before the last 25 years doing this, which immediately means 50 years of doing work, which I don't like at all. Days like this say one thing to me: Florida. Why am I not there? So the idea is that I try to make this, I probably dedicate the first half of this class to kind of the anatomy and physiology of microorganisms, understanding that, understanding the different kinds that they are, how they grow, different ways that we might get rid of them. Okay, all that type of stuff. A quick email I got to respond to. Hold on. Don't have children. I already said that before, despite all three of mine. And so I think it's really important to get that part done because that's an important part for you as well. And then the second half of it, I try to do mostly about diseases, how they affect people, how they're transmitted. Because if you're going into a health career, yeah, and even if you're not, It's good. It's certainly useful information. If you're going to a health oriented, you want to be able to know if you have somebody who has an acute respiratory disease, let's say an acute RSV, which is going around right now, respiratory syncytial virus, which we'll learn about it, which is potentially life-threatening. Not only what precautions you have to take so that they don't get worse, what you have to do to not spread it, what you have to do to not get it or contaminate somebody else or bring it to other. I mean, all of that's part of this to me. That's there. So that's really the jumping off point for this. From an exam perspective, we typically have 4 exams that are objective in nature, which means multiple choice, true, false, matching, that type of stuff. That's called a recognition test. You see the answers, you just got to pick out the right one. Okay, that's recognition. In the lab, you'll be utilizing much more of subjective measures, doing activities, writing reports. That's subjective in nature. I don't do the essay and short answer questions. Why don't I do that? Is because the vast majority of the students who are in health or nursing have to pass a licensure exam. And that licensure exam is objective in nature. And there is literally no way to cheat. None. If you have never been, uh, if you have never seen the mechanics of these things, whether it's what we were teaching earlier this morning at 10 for the pre-med students preparing for the MCATs, or any of these other things, or the NCLEX or anything like that, basically you walk into a room through metal detectors and they'll have somebody there scanning you if you go off to make sure you don't have anything electronic with you. They'll give you a pencil and some scratch paper, which they will take away at the very end, or when you used up the paper, give you another piece. You're in an isolated environment, walled off, where you can't see or hear anybody else, and it's you versus the computer. There's no way to cheat. And plenty of people cheat on other things. It's very hard to do that. And I'm fond of saying you only cheat yourself when you're doing it. So understand it. That's why I do these objective tests. You have to get used to dealing with high level, difficult objective questions. Let me decide on what the value of your grade is. I'd rather make it hard so you're effectively practicing for the real thing. And so maybe you get 80 or 85% correct. That might be awful for some, because I always get that. I need to get at least an 80. Oh, I don't want to hear it. Okay. Effectively, we don't do number grades, we do letter grades. What the nursing department or other departments do is not my concern. And moreover, so if you get a 70%, my guess is it's probably going to be a grade that's in the B, at the very least a high C level, based on my experience. Because I know, having been doing this teaching basically nursing and health career majors for the last, 20 plus years, how performance on my tests relate to potential success in your field of endeavor. So you're going to have to rely on my experience. And if you ask anybody, I test hard, grade relatively easily. Would you say that's a fair assessment? Okay. Unless I don't like you and I'll grade, I'll do both. Test hard and grade. I didn't mean that. I was joking. They laughed. You guys you're already that side of the room, strike one. Haven't they told? It's the same tired old jokes I've done for 20 years. Same stupid anecdotes, same stories, pictures of my kids, pictures of my grandkids, pictures of the dog, pictures of the cats, the whole thing. It's all we get. It's just, if anybody asks you, just say he's old. See, and none of you had to agree. Now you're in trouble. You go, yeah, you're old. Ugh. Why do I do this to myself? I could be retired. I should be. Sorry. Moving on. Okay. Oh, back to the syllabus. That's what I was jotted away from. I never remember this stuff. Okay. And so, textbook. They're doing it differently these days with these textbook bundles. The major issue, look, if you want to buy a text, buy a text. If you don't. If you're getting it as a bundle, you get it. The issue is the software package, which they call Connect. I don't know if it comes with it or not. I'm sure it's acquirable. I don't use it. It's like when you had A and P, for some of you, you had that mastering thing. A lot of instructors want that. They do homework through it. They do it. I don't use it. Like, I mean, you're in college. It's on you to do this. I'm not a homework. We don't need work products. We need performance. Okay. So it's all about performance related. So if you want, I don't mind putting together modules if you want me to. And I've had students ask me that. I've set up a site. If I didn't send it to you, I'll send you the logon information. Effectively, what happens to software that accompanies the text, it used to be free with McGraw-Hill. It was wonderful. Okay. And, it's all about, there's a certain amount of profit. So as they went away from hardback textbooks, which were very pricey, to e-textbooks and notebook style textbooks, which are not as pricey to produce, they decided to make some money on the software. So it transitioned out 6 or 7 years ago. You could just log on for free and have a lot of animations and questions and test prep and stuff, and modules that you can. If you like them, just let me know. I'll be happy to put one together for you. It's my job, but it's not a requirement. You have to choose what you utilize based on your budget if it's an applicable concern. It costs a lot of money. I know that. I am fortunate that only my eldest went to college and he went on a full scholarship. So Uncle Sam paid for that one. So I was pretty happy with that. Okay. So the exams, we scale them. It converts to a letter grade. If you have questions, we'll go over it, I'm sure, again. I don't want to waste any more time on that. And the rest of this is pretty straightforward. Okay. And the exams will be announced. The exams will have a study guide. I will try to emphasize the points. I'll record a study guide and distribute it. I will go over it. I'm happy to answer questions in class. You can email me anytime. I have nothing better to do than respond to emails and watch sporting events. And with the Steelers eliminated, I have entirely too much time. But it's okay. We made the playoffs. Hey, I'm good. I'm good with it. Now now I have to go full blown on the Penguins, and it's going to be an effort to get into play. Very competitive. This is roughly the topics that we're doing in the exam dates and the contents, and I'll go over the blackboard. And I'm sure you've seen this already. This is the required information that the powers that be, and it's fine. And like any of these policies, they're pretty much self-explanatory. There may be times when we need to go remote if you don't feel good. That's why you can stay in your room or at home, however whether you're a commuter or you're residing here during the term and interact. Any of those things are possible. Okay. So we're trying to get, all this other stuff, academic, and I try to not stay involved with that if I can help. It's just, to me, the acid test is you understanding and learning the material, because I think it's important to you and to anybody you're going to interact with in the future. Which brings us to the website, to the Blackboard site. So, and I've preloaded it. Unfortunately, I've loaded it a couple of times, so there's duplication. But effectively, test 1 is week 1, test 2 is week 2, week 3, test 3, week 4, the final. Some modifications. We may use the other sites. I may put other stuff on there. All of the PowerPoints, plus PowerPoints we've used previously for the prior dozen or more years, we used this textbook called Nestor, which I will go back to because I like some of the illustrations and I'm comfortable with it, having used it for 15 years at a couple of different schools. I've used this Cowan text multiple times. I taught at CCAC at Westmoreland Community College, even when I was teaching here, and they used the Cowan text. So it's a good text, and they're both McGraw-Hill texts. It's like, for instance, we get to the famous Pasteur experiment. We'll use Nestor because I like the slides better. And so I go back and forth and I put everything on there, plus outlines if I have chapters that are outlined, and some of them are. I would say the majority are. Animations, which I have as a zip file, different animations, which I pulled out specifically. These are old YouTubes, which you can always go back and look at one. You can see the dates. That was 120 of last year, because last time I taught this was last year. There's interesting stuff to look at. That's an animation that we'll be looking at here. So they're there. And I have different outlines and different presentations. So what I'm going to try to do, not to confuse you, is to send your YouTubes to you by way of Blackboard, like the one I sent out earlier today. So what I sent out earlier today to this D was this. So I sent them that. That was the YouTube I sent them. So I converted this Google Meets to a YouTube. It's a little more user friendly. Oh, good. 16 minutes. Let's get started. Okay, you ready? This stuff's not easy. I want to be very sincere with everybody. Of the core sciences for people in nursing and health careers, this typically is the most difficult course, more so than anatomy and physiology, I think. Not always. Depends on who's the instructor. There's a lot, because it's unfamiliar territory. I mean, microbiology, first of all, it's a misnomer. It's the wrong name. It should be microbes, study of microbes. Because biology, definition, ology means study of, bio means life. So micro means stuff you can't see. Microbiology, study of living things you can't see. Viruses aren't living. Okay, so immediately it's something called prions, which means they have cow disease. And other diseases are also not living because they're not made of cells, and living things are made out of cells. So there's a lot of this builds on what you all saw in anatomy and physiology. What makes a living thing? Okay? It's got DNA. It's made out of cells. It's got DNA and RNA. Those don't. Those might have one or the other, or might not even have any of them. Prions are just proteins. Viruses are either DNA or RNA, not both. It's got some type of membrane around it. That doesn't necessarily apply to viruses or prions, so they don't meet any of those tests. Living things do energy transformations like we do. We eat something, we convert it to energy or building blocks, and we excrete. That's energy transformations. So bacteria do that, and really our focus point for this class is bacteria and viruses, more than anything else. There are things you encounter the most, there are things that affect us. We also do other important disease-causing and other organisms that are important vis-a-vis us. And those are what are called eukaryotic organisms. We'll get to that in a second. Eukarya are what we are. We have cells, membrane-bound organisms, except we're multicellular. We've got billions, trillions of cells. Whereas prokarya, you'll see the definition shortly, are single-celled organisms. That's what bacteria are. They're there, and you're going to get viruses, already made cells. They're there, but there's a lot of, and really, they're all important. Yeasts and fungi are all eukaryotic organisms, something called protozoans. Protozoans, we don't think about. It's like when you were in high school, you had pond water, you looked into the microscope, you saw all those things spinning, swimming and moving around, you know what I'm talking about? That kind of stuff. Those are called protists, or prokaryotes. They're organisms that are similar to ours, but they're typically a single cell. And, well, what's the big deal? They're in water, and as long as we're careful with the water we drink, we don't get sick from that because they can contaminate. They do. We ever know somebody with well water is contaminated. We'll talk about that as well. But the reality is that those diseases also caused by them are significant. The leading cause we did not know of death in sub-Saharan or equatorial Africa. And most people get this wrong, think it's HIV/AIDS. It's not. Malaria. Malaria is caused by a protozoa that affects the blood. Then HIV/AIDS. Then another viral disease you might have heard of, obesity. Surprises me. We'll learn more about that when we get to it. So there's a lot that goes on with this particular area. So we have to go over the anatomy and physiology and definitions. And the hard part to me is we're dealing with a cell, which most of you are not familiar with. The bacterial structure is what's called a prokaryotic cell. It doesn't have a nucleus. It doesn't have membrane-bound organelles. You learn A and P. First thing you learn in AP1, membrane-bound organelles. You got a nucleus. You got mitochondria. We got lysosomes and peroxisomes and roughly smooth endoplasmic. Right? Heard all that stuff before? Not here, thank you very much. Okay. All you got is a cell with a cell wall. Bacteria all have cell walls. Not necessarily all form the same way. And that's an important distinction. That may be the single most important part of the first test. Something to do in lab. It's called gram-positive versus gram-negative. It's not the unit of measure of gram, like milligrams or kilograms. It's a proper name. They give the doctor whose last name is Gram. Danish. Capital Gram. And there's two types of structures: gram-positive, like staph and strep infection, and gram-negatives, like E. coli and so on. And they're totally different. And we have to treat them differently, but they're both bacteria. It's all based on their cell wall. That's a really important distinction. So understand that those things don't have a nucleus. They've got DNA, and the DNA is not in, we all know our DNA, we're what's called diploid, pairs of chromosomes. Called 23andMe, Ancestry.com. That, right? People have done that, they're into that stuff, I'm not. See, I mean, other than the X and Y chromosomes, we've got 22 others that do whatever they called autosomes, and that are our characteristics. Bacteria have a ton of chromosomes. They're all in one big circle. So it's one big chromosome. They have a ton of genes in one big circle, and all that is like a giant rubber band that you keep twisting and twisting and folding and twisting. It's called super compact. It's all squeezed into this teeny tiny cell. You'll see some illustrations. And it's got some ribosomes to make proteins, because you have to be able to have DNA and RNA. That's a living thing. You have a cell membrane, but it's got a cell wall around it, and that's about it. Not a whole lot else going on. The big thing that hurts us with both viruses and bacteria is they are geared to do one thing. Everything that they are made of is for one thing: replicate. Making very simple. They don't do mitosis or meiosis or anything like that. All they do is make more. And they do it extraordinarily rapidly. And in a matter of hours, you can go from one to literally millions of things like bacteria or viruses. So they replicate exponentially rapidly. So the bottom line is, if you're infected by one of them, and you don't do something about it now, no question about it. We'll talk about something most of you have been immunized for, whether they still required or not around. We'll get into that later. No requirement of immunization. Meningitis, right? Ringing a bell? Does that have a meningeal immunization? Yes, no, so. Yeah. Okay. Bacterial meningitis, if you don't make the diagnosis and start treating it now, somebody's going to die fast. Viral meningitis is not necessarily as fast. You have a little bit of what, for lack of a better way to describe, wiggle room. You bring a child, I tell us all the time, to Children's Hospital. This was the protocol years ago, and whether they changed it or not, 15 or 20 years ago. What's called fever of unknown origin. Okay, 104. That 104 fever might get me or you into the hospital. It's not a ticket to get admission for a child, because that's children get out. If you don't know what's causing it, here's how they go about it. First test, they capture urine, which is easy to do and non-invasive. You don't have to start sticking people for blood tests or taking saliva or any of that stuff. You see if there's a bacterial effect, because surely an infant can't tell you very easily. If that doesn't give you the answer, well, they better do blood tests. No, chest X-ray. Baby screaming and yelling is very hard to hear. Respiratory sounds, chest X-ray. Let's see if it's normal. If you don't have an answer, then they still haven't done the blood tests. The next test is a lumbar puncture or spinal. Most of you learned about in AP1. Because by that time, after those, maybe an hour or so of those two tests, you better find out now whether they've got meningitis. And that's the diagnostic test. Spinal tap to see if you see evidence of bacteria or white blood cells through the spinal fluid. Period. Now, at the same time, we should do blood tests and other stuff. And it's scary when that happens. I'll recount that story because it happened to my son. Not the one who built the car, the other one. All those many years ago. He's 40. See, somebody could say, you can't have a 40-year-old son. But no, you all just sat there like, why he's not 50? I digress. Okay. You're not going to laugh at the jokes. You're in trouble. Let us begin. So that's the introduction. So, okay. Things we can't see. And they say living things. And again, viruses, which are either have DNA or RNA, not both. Prions, proteins only, that's where the PR comes from. All the rest of these have impact to us. And by and large, we consider them in this course. Bacteria, certainly. Archaea, or archaea, depending on how you pronounce it, derivative are very old. They're derived from the word like archaeology. They are bacteria-like, but not identical, and we'll go over the criteria which distinguishes them. By far and away, the most important one is that they only exist in extreme environments. Volcanic caldera, bottom of, polar ice pack, bottom of the ocean, stuff like that. Okay, so we don't really interact with them very much. Bacteria kind of live everywhere, some even in extreme environments. But archaea are interesting research-wise, particularly on how they've survived so long and adaptations and those. So there's more interest in them today than there used to be. But bacteria is a very significant part of this course. Protozoans I just talked about, like the pond water samples. Protozoan, fungi, helminths, which are worms, and algae are all eukaryotic. Their cells are like ours. It's good news, bad news. We understand their cells because they're like ours. The trouble is, if you want to treat a protozoan or a fungal infection or a worm infection, you have to use medication that will destroy that cell. It's like ours. So there's a greater chance of side effects or collateral damage because it's attacking a cell that's very much like ours. That's always the problem. For years, we'd had very, very little effective oral antifungal medication. Now we have better medications for that. So it's understand there's a lot of, and we get into antivirals, antibiotics, and fungals, which you'll find very helpful. Helminths are microscopic when they start as a single cell, but they aggregate and form worms. To me, they're more macroscopic. So I just talk about them in passing because I don't understand them that well, and really they're basically macroscopic. If you're interested, if you know somebody who is a veterinarian or works with that, they understand it very well because it certainly is a significant feature with regard to our animals and particularly our pet animal population. Our dog takes heartworm medication, the whole flea and tick collars and all that type of stuff. Algae are water-based, and it could happen in your pool outside or in the ocean. They are eukaryotic, so their cells are like ours. The novel thing, and if they're the only ones that have this, they have chlorophyll and they have a cell wall. So they are very important, particularly with regard to our own lives, because they are photosynthetic organisms, because they have chlorophyll, like plants. You'll notice plants and animals aren't in here because they're too big. They're not by definition microscopic. Yes, the cell structures are in there, but not the same thing. The only one that is photosynthetic like a plant are algae. They are responsible for the vast majority of our oxygen. Our oxygen on this planet comes from photosynthesis, which relies on chlorophyll, which relies on plants and algae. Our surface of our planet is about 5/6 water. The oxygen comes predominantly, vast majority, from algae. If you're going to say here, we get it a lot from the plants that are here. Even in winter, we still have sufficient amount of greenery and chlorophyll, and a little bit in our water supply. But again, we're mostly a water-covered planet that are there, and that's a unique feature. Unique is an important word. Unique doesn't mean special or distinctive. In a science classroom and in biology, unique means something that no one else has. So eukaryotic organisms, other than plants, the only microscopic ones, algae. And we'll see the same thing applies when we start talking about bacteria. They contain a magical substance that nothing else has, called peptidoglycan. Peptidoglycan distinguishes bacteria, and more importantly, because it's in bacteria, not in us, it's a target for us to attack with antibiotics. The single most important target. Okay. And the antibiotic family that attacks it start with something called penicillin, which you've heard of. And we'll get more about that because we'll do a whole unit about that. Okay, so they're easy, and you'll see this in the laboratory, because you can put a little Q-tips worth on a plate and incubate it and come back a week later and that plate is covered with bacteria. So we can study it. We can't see them without using the microscope, but we can see features that help distinguish it, and certainly microscopically and chemicals that are used to test it. So typically the way we test to see if it's Staphylococcus or Streptococcus, as you'll do in the laboratory, most of you, my colleague, Dr. Dress, who, despite me telling you, is a very nice guy. So you'll have a good time. I don't know who the other instructor is. And what you will do is you will stain these, and it will reveal what category of bacteria it is. So we have to do things indirectly because they're very, very hard to see. Okay. And a lot of times when they say indirect, well, one of the tests we use a lot is we use fermentation tests. Certain bacteria only gobble up certain nutrients, and typically with those nutrients, they ferment them. So if you take a sugar and ferment it, you make some kind of an acid. And then we use pH indicators to turn colors, and that's easy. If it goes from red to yellow, it's gobbling up. It gobbles up a certain sugar, but I don't want to do lab. So by definition, mushrooms, which we see with the naked eye, are not microbes. Microbe is a better term than microbiology because microbe is more global. When you say microbiology or studying, because bacteria are living again, viruses are not. So they've been around. I'm not getting into evolutionary biology. This is the practical science part of it. People have different views about how these came on, but this is the scientific view, which I teach. Okay, single-celled organisms based on carbon dating about 3.8 billion years ago. No, I was not here then. You're probably around for that. Okay. Single ancestor. We're going to talk about that because there's a very important theory. Theories in science are pretty close to facts. There's basically people trying to punch holes in them and they can't. It's called endosymbiotic theory. It's kind of how, like, our cells evolved from precursor cells, some of which were bacterial. So eukaryotes, U for true or with, have the carrion as the core. So with a nucleus, the cell you studied in A and P, okay, membrane-bound organelles is really a better way to describe it. Bacteria, no nucleus, they call it a nucleoid. Looks like a nucleus, but it's not. Archaea, same thing, but distinct from bacteria because of where they live and some other unusual findings, in particular their cell wall and cell membrane structure, to accommodate those extreme environments. Prokarya is the global term for both of these. Okay, so both bacteria and archaea are in the same basic large grouping. And, I've never used that term, so let's skip it. And this is really, if we want to think at living things from a common ancestor, this is sort of a construct for the domain system. There are 3 domains, 2 prokaryotic, single cell, cell wall, bacteria living everywhere, a lot of different types. Archaea, living in extreme environments, a lot of different types based on their metabolism. And then eukarya, us, as well as plants, animals, slime molds, and we classify them based on ways that they move or certain structural things that are. And we will get in, I have a table constructed a little bit better than this illustration. And as you'll see on the blackboard, I have some of the stuff already printed out. During COVID, when we were remote a lot, we really didn't have a blackboard to write on. So you typed it out and I just distributed it. They're not predominantly single cell. They're all single cell. They all have cell wall, and they're really small. Eukarya, mostly single cell, but can be multi-cell like us. And so they can become highly complex. In general, they're larger. Eukaryotic cells, in the lab, when you look at yeast, you'll look at it under high dry. When you look at bacteria, you have to look at it under oil immersion. The difference between that is 2.5 times. High dry is 400 magnification. Oil immersion, which you do a lot of, is 1,000. So their eukarya are considerably larger. And they give you a chart about when things developed. And bacteria and archaea, more or less, whenever from the common ancestor, then eukarya, and then the evolution of that. And they are everywhere. Every square nanometer on your skin is covered in microorganisms. You can go wash your hands. You can come back in here and hit the hand sanitizer, and I can test your skin, and it will have microorganisms on it. It just will. You can't get rid of them. They are everywhere. Okay? They are your friends. They get a bad reputation. Oh my God, you have bacteria on there. They cover up sites so the bad guys don't have a place to land and live and flourish. So the vast, vast, vast majority are not what we call pathogenic or disease causing. They're your buddies. So on your skin, you have different kinds of bacteria and different kinds of yeast, and they don't trouble us whatsoever. If anything, it's helpful because your immune system is sampling the environment all the time. And what it's doing is basically keeping itself in tune. Oh, I saw that. That's okay. We can let it go. Uh-oh, there's a problem. We better address it right now. So it's a use it or lose it type of a thing. We'll get into something called the hygiene hypothesis and why that's problematic. And unfortunately, a lot of the problems were magnified by a variety of reasons, not the least of which was this immense lockdown that was frankly ill-advised during COVID. And the people I feel the worst for are you guys. It was easy for me. My wife and I got to stay at home the whole time with the animals and teach from there. It was fun. Okay? Had a good time. Yeah, you had to learn remotely. When you came back, you had to be masked. And awful. I mean, so a lot. I think it's going to be impactful on many folks of your generation. That's my own opinion. I don't have any scientific basis for this, but I really think it's a problem. So they're everywhere. Think of any environment, we have microbial forms. Even inside of us, if whatever your termination is, we carry them. A lot of animals carry diseases that don't affect them. Like when you hear about things like Lyme disease, which is a tick-borne disease, the tick's not suffering from it, okay? The tick's not dying from it. The tick is called a biological vector. It lives in their bloodstream, and then when it seeks a blood meal and we happen to be in proximity, you can get Lyme disease. And Lyme disease is the number one vector-borne, meaning being carried by another species to us, number one vector-borne disease in this country, and not insignificant whatsoever. and for us as well as for pets and things like that. So they're everywhere. And the essentiality was what I was telling you about the oxygen and other things. Photosynthesis is a means of doing what's called carbon fixation. When you fit, it's not repair in the sense of fix something like a broken window. It means to affix. It's taking inorganic carbon, carbon dioxide, and sticking and creating bonds and bonding it together to create hydrocarbons, organic compounds, all the things you learned about in AP1, proteins and carbohydrates and fats and nucleotides, right? So we need bacteria as well as other photosynthetic organisms for that. Bacteria are responsible for fixing nitrogen. Our atmosphere is 80% nitrogen gas, 20% oxygen. That nitrogen gas is how we fix or attach nitrogen or nitrogen compounds to other organic molecules like amino acids or nucleic acid bases, which are essential for life. So bacteria allow us to do that. So they're essential in that regard. And there's just, and you'll see a whole laundry list of important things they do. Bioremediation, we manipulate bacteria to get rid of oil spills. They're in the food industry, they're everywhere. My personal favorite. Okay. For you folks, for the people who like alcoholic beverages, where would you be without yeast? For me, I'm thinking of sourdough bread. Not a value judgment. So again, scientific theories are things that people try to poke holes in. They're just about this side of a law, like Newton's law. So we see it all the time. They're easily testable, years and years, blah, 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 blah. Is what not, oh, baloney. Photosynthesis, carbon, oxygen. It's the only one you really have to know. I'm not worried about anoxygenic or oxygenic. Okay. We'll get into that when we do energy transformations in microorganisms. Again, carbon dioxide, nitric oxide, an important substance. Methane, again, important substances, all of which are a bit more atmospheric in nature. Bacteria in the oceans, that's where the oxygen. Viruses are also present, so they're everywhere. There are things. So are found everywhere on the planet, not except they're everywhere. So that's what, when you see questions phrased like that, usually they're struggling to make it's like a true false. It's easy to make truths, it's hard to make falses. So you got when you become a more expert test taker, it's a little easier to pick out falses than truths. I think it certainly make the questions. Genetic engineering. Every time you buy a product that says GMO, or even for that matter says non-GMO, it tells you the concern that we have. We take bacteria like E. coli, we take eukaryotic organisms, mostly yeast, the same yeast that you do for baking and brewing, Saccharomyces cerevisiae, the same one you get a yeast packet when you're doing a recipe at home. Okay? And because they're relatively simple organisms that you can play with, we insert DNA into them and resequence effectively their DNA, reprogram it. So it makes a different protein or it develops different characteristics. Okay, that's genetically modified. Sometimes they're for good, sometimes they're for bad. I mean, if you know someone who's a diabetic and is insulin dependent, that's where their insulin comes from. The first genetically modified product in 1977 was insulin. It originally was called humulin or human insulin. If you did not know, the original source of insulin was from pigs. They would grind up pig pancreas where it's produced and extract it. And a lot of people either were allergic to it or didn't want to use it because of religious reasons or whatever. Okay. Then they switched it to bovine insulin. When I was in medical school, it was bovine insulin. And the same thing, when cows were slaughtered, they'd grind up the pancreas, extract the insulin, and that was used extensively. But again, same subsets from a foreign product, there's going to be allergies. It's not yours. So they analyzed the genetic code and the proteins, the sequences that was known, and basically you constructed a DNA strand that you could insert, and we'll go over how that is done by a neat thing in bacteria called plasmids. It's the way it can shift DNA from one to another. Bacteria are great like that. I mean, a bacteria, let's say the bacteria here has 20/20 vision, and all the rest of us who have glasses or don't have 20/20 vision, all she would have to do is touch us, and she could magically make all of our eyesight problems disappear. We can't do that. Bacteria can do that. So much so that one MRSA could change an entire population of Staphylococcus to MRSA overnight. One. So that, and we use, this is all from what's called recombinant DNA technology. We'll talk a little bit about that. Bioremediation is why I was gobbling up oil spills, eating up toxic pollutants, et cetera, et cetera. It's a good thing. Thanks. Be a little extra in your paycheck. And then associated with humans are harmless or beneficial. There's really only about 2,000 that cause disease. There's hundreds of thousands, but there's so many infections. Now, infectious diseases are important and common causes of death worldwide. They probably have the thing. And I didn't look at this with the other class, and I want to look at it and explain it to you, so I'm going to stop here. But because I spent a little less time doing the introductory stuff, is to understand that infectious disease is by no means eradicated, and that we still have a tremendous amount of problems with it. So thanks very much. We'll see everybody on Friday, and I'll distribute this to you later, either tonight